The overall goal of this procedure is to set up a functional TetON system in Zebrafish to achieve conditional tissue-specific gene expression, as demonstrated in the regenerating tail fin in this video. This is accomplished by first generating TET activator transgenic lines that express a tetracycline-inducible transcriptional activator tagged with AmCyan in the tissue of interest, and TET responder lines that contain a YPET tagged gene of interest under control of TET-responsive elements. Next, specific combinations of TET activator, TET responder double transgenic fish are established. Then, tissue-specific TET responder transgene expression is induced through administration of doxycycline. Finally, TET responder transgene expression is verified by preparing tissue sections, here of fin regenerates. Ultimately, immunofluorescence is used to visualize tissue-specific TET responder transgene expression.
The main advantage of this technique over other methods, such as heat shock-induced gene expression, is that the TetON system allows for inducible and reversible gene expression in a tissue-specific manner.
This method can help to gain a more profound understanding of the molecular signaling networks guiding zebrafish fin regeneration, as it allows for analysis of signaling pathway and gene function in individual tissues.
While we use this technology to study zebrafish fin regeneration, it can be instrumental also in many other contexts requiring tissue-specific manipulation of gene function.
After generating TET responder fish lines according to the text protocol, assess for